Welcome to Medicalink. Don't forget to subscribe for more videos. Let's learn.
Hello everyone. Today we'll be discussing on vitamin B12. This vitamin B12 acts as a cofactor for various enzymes. We'll talk about those enzymes. We'll talk about the metabolic roles of vitamin B12 and the deficiency of vitamin B12. So basically vitamin B12 is a cobalt containing compound and it has corrin ring, and the central portion of vitamin B12 consists of 4 pyrrole rings. So what are the vitamin B12 dependent enzymes? So vitamin B12 dependent enzymes are methyl malonyl coenzyme mutase. It is methyl malonyl coenzyme mutase, leucine amino mutase and methionine synthase. So methyl malonyl coenzyme mutase, methionine synthase and leucine amino mutase are vitamin B12 dependent enzymes. And vitamin B12 acts as a cofactor for various enzymes. So what is a cofactor? So cofactor is a protein chemical compound, or it can be metallic ion, which is required for an enzyme activity to act as a catalyst. So it is a helper molecule of enzyme. So vitamin B12 is a cofactor for methyl malonyl coenzyme A mutase, methionine synthase and homocysteine methyl transferase, where methyl malonyl coenzyme A mutase will convert methyl malonyl coenzyme A to succinyl coenzyme A, and methionine synthase causes remethylation of homocysteine to methionine, and homocysteine methyl transferase causes transfer of methyl group as methyl cobalamin. Now coming to vitamin B12 and homocysteine metabolism. Methionine synthase is a vitamin B12 dependent enzyme that catalyzes the formation of methionine from homocysteine using 5-methyl tetrahydrofolate. This 5-methyl tetrahydrofolate will act as a methyl donor. Methyl group is transferred from 5-methyl tetrahydrofolate to vitamin B12, forming methylated vitamin B12. So methionine synthase will cause transfer of methyl group from 5-methyl tetrahydrofolate to vitamin B12 to form methylated vitamin B12, and during the process 5-methyl tetrahydrofolate will get converted into tetrahydrofolate after losing its methyl group in the presence of the enzyme methionine synthase. Here tetrahydrofolate has various forms. So tetrahydrofolate can get converted into N10-formyl tetrahydrofolate, which is the most oxidized form of tetrahydrofolate. It can also get converted into N10-methylene tetrahydrofolate and N5-methyl tetrahydrofolate. N5-methyl tetrahydrofolate is the most reduced form, and they are required for DNA replication, helping in DNA synthesis. Now methylated vitamin B12 will then transfer its methyl group to homocysteine, and this homocysteine will get converted into methionine in the presence of the enzyme methionine synthase. And methionine is required for cell proliferation, protein synthesis, RNA synthesis and transamination. Now methionine will get converted into s-adenosyl methionine, also written as SAM, that is s-adenosyl methionine. S-adenosyl methionine helps in methylation of DNA, RNA, protein and phospholipids. This s-adenosyl methionine also helps in synthesis of polyamines. When acting as a methyl donor, s-adenosyl methionine forms homocysteine, which may be remethylated by methylene tetrahydrofolate catalyzed by methionine synthase. Methionine synthase uses 5-methyl tetrahydrofolate as a substrate. Thus, indirectly, it helps in maintaining the level of tetrahydrofolate. Now, in case of vitamin B12 deficiency, the activity of methionine synthase will decrease. The activity of methionine synthase will decrease. So, when the methionine synthase activity is decreased, 5-methyl tetrahydrofolate will not get converted into tetrahydrofolate, because 5-methyl tetrahydrofolate will not be able to transfer the methyl group to vitamin B12. So, in this case, methionine is not formed from homocysteine. So, there will be decrease in the level of tetrahydrofolate, and there will be increase in 5-methyl tetrahydrofolate. There will be increase in 5-methyl tetrahydrofolate, and since homocysteine is not getting converted into methionine, the concentration of homocysteine will also increase, and there will be decrease in methionine concentration, and methionine will not be formed, and s-adenosyl methionine will also decrease, and the level of s-adenosyl methionine will also decrease. And because homocysteine is not getting converted into methionine due to reduced activity of methionine synthase in vitamin B12 deficiency, leads to increased concentration of homocysteine. So when there is rise in homocysteine level, it causes endothelial damage, and the patient will present with cardiovascular manifestation. Similarly, homocysteine is not getting converted into methionine, so the level of methionine will be reduced. So there will be decreased cell proliferation, there will be reduced protein synthesis, RNA synthesis, and transamination. So cell proliferation, protein synthesis, RNA synthesis, and transamination will be affected. And because there is reduced level of methionine, methionine is not getting converted into s-adenosyl methionine, which is important for methylation of DNA, RNA, protein, and phospholipids. And there will be defective synthesis of polyamines. Because of the impairment of methionine synthase in vitamin B12 deficiency, this will result in the accumulation of 5-methyl tetrahydrofolate that cannot be used. So this is known as folate trap, where there is functional deficiency of folate secondary to vitamin B12 deficiency. Now we'll talk about the folate trap hypothesis. So the effect of cobalamin deficiency are more frequently seen in rapidly dividing cells such as erythropoietic tissue of bone marrow and mucosal cells of the intestine. Such tissue needs both N10-methylene tetrahydrofolate and N10-formyl tetrahydrofolate for synthesis of nucleotide, which is required for DNA replication, thus helping in DNA synthesis. However, in vitamin B12 deficiency, utilization of N5-methyl form of tetrahydrofolate in conversion of homocysteine to methionine is impaired. And because the methylated form cannot be converted directly to other forms of tetrahydrofolate, so folate gets trapped in N5-methyl form of tetrahydrofolate, which gets accumulated, and the level of other forms of tetrahydrofolate will decrease. So methylated tetrahydrofolate is not getting converted into tetrahydrofolate, and this will lead to defect in rapidly dividing cells of erythropoietic tissue of bone marrow, leading to megaloblastic anemia. So this will lead to megaloblastic anemia. Vitamin B12 also acts as a cofactor for L-methyl-malonyl coenzyme A mutase enzyme. Degradation of propionate is an essential step in lipid and protein metabolism. During the degradation of propionate, L-methyl-malonyl coenzyme A gets converted into succinyl coenzyme A in the presence of the enzyme methylmalonyl coenzyme A mutase, and this requires vitamin B12 as a cofactor. The resultant compound succinyl coenzyme A will enter into Krebs cycle, and succinyl coenzyme A is essential for heme synthesis as well. So when there is deficiency of vitamin B12, the activity of methylmalonyl coenzyme A mutase will be greatly reduced. This will lead to accumulation of L-methylmalonyl coenzyme A. When there is increased level of L-methylmalonyl coenzyme A, this will cause urinary excretion of methylmalonic acid. When there is increased concentration of L-methylmalonyl coenzyme A, there will be urinary excretion of methylmalonic acid. This will provide a mean of assessing vitamin B12 nutritional status. Vitamin B12 is synthesized exclusively by microorganisms, and they are found in foods of animal origin, and it is absorbed from distal 1/3 of the ileum. Now we will talk about metabolic roles of vitamin B12. So vitamin B12 causes isomerization of methylmalonyl coenzyme A to succinyl coenzyme A by methylmalonyl coenzyme A mutase. It also plays a role in methylation of homocysteine to methionine by homocysteine methyltransferase. Vitamin B12 also has a role in methylation of pyrimidine ring to form thiamine and metabolism of diols. In bacteria, vitamin B12 causes interconversion of glutamate and beta-methyl aspartate. Now coming to deficiency of vitamin B12, vitamin B12 deficiency can cause megaloblastic anemia. Beside megaloblastic anemia, it causes methylmalonic aciduria. As we said, methylmalonyl coenzyme A mutase is a vitamin B12 dependent enzyme, and in deficiency of vitamin B12, it leads to methylmalonic aciduria as it is excreted in the urine. Deficiency of vitamin B12 can also cause folate trap and pernicious anemia. It may lead to subacute combined degeneration and demyelination of spinal cord. Knuckle hyperpigmentation may also be a common manifestation of vitamin B12 deficiency. Other causes of knuckle hyperpigmentation beside vitamin B12 deficiency are hemochromatosis, Addison's disease, and thyroid disorder. Thank you.